students, I am RG ma'am is with you to start the lecture number 7 from now. Okay, till now we have read about what is cell organelle, what is cell wall, what is cell membrane and everything else about cell. Now there is a mystery. How can we see the cell? Do we need some kind of structure or instrument to see the cell? So the basic thing that we need to see the cell is the microscope. So microscope is the basic instrument through which each and every cell can be observed. Okay, we can see the smaller and the smallest part of the cell itself. Even the cell organelles can be seen clearly under the microscope. So in today's lecture, we are going to start about microscope, what is microscope, who discovered it, types of microscopes and many more. And the great interesting thing which I am going to tell you about this lecture is we are going to see good videos about our cell organelles till which we have read till now. Okay, the cell organelles like chloroplast, ribosome, lysosome, endoplasmic reticulum and many more. And the last part that was cell division which I told you about that in the lecture number 6. Okay, so I am going to start my lecture today. So let us see the basic contents of our lecture first. So you know very well what is cell. Now we are going to see the content of this lecture. That is, first one is the microscope, the basic thing or the basic instrument used for visualization of all the small cell organelles. And the last and the very interesting part we are going to see is video study of the cell organelles. Okay, so let us start with the microscope. So what is microscope, student? It is an instrument used to study the various cell organelles, or we can say anything which cannot be seen by our naked eyes is visualized under microscope. Means the things which have very less diameter or they are very small in size, they can be seen under microscope. They cannot be seen through our naked eyes. Okay, now there is a great preparation to see any kind of cell inside the microscope. Okay, so I will tell you it further. First we have to see who discovered it. So the names are written over here. The discoverers were F. Jensen and Zacharias Jensen. The first compound microscope was built by both of them, F. Jensen and Zacharias Jensen, in 1590. So you just have to remember the name of the scientist as well, that who discovered the first compound microscope, F. Jensen and Zacharias Jensen. Okay, now moving towards the next slide, we are going to see that how the structure of the microscope is being organized. So first you have to see, students, in our schools we see the microscope. Okay, microscope, you see, it is the compound microscope in which all the cell organelles are observed. Okay, now what are the parts of the microscope? First is the base part. It is the basal part of the microscope which carries on all the weight of the microscope. Okay, so it is also written over here. It bears the whole weight of the microscope. Okay, we will see it through a diagram also, but first we should see all the parts of it. Okay, the second part is the handle part. The handle part, this part is handle part. You can see it in my slide also. This part is known as handle part of the microscope. Okay, it is a curved part. Okay, it is not flattened, it is just curved part. Next part is the stage. It is a strong metallic rectangular structure which is horizontal plate fixed to the handle. Okay, this type of structure on which the lenses are being fixed is known as, here you can see here, it is the stage. Okay, now next part is the stage clips. The stage clips are used to fix the slide which has been prepared to see under the microscope. Okay, now next part is condenser. Condenser is the basic part of the microscope. Now why it is the basic part, students? The main important thing about that is it is used for concentrating the light angles. Okay, now moving on towards the next slide, we will see here the body tube. What is the body tube? It is a wide hollow structure hollow tube attached to the upper part of the arm, and this tube lenses are attached. Okay, the lenses are attached over the body tube, students. Next part of microscope is adjustment screw. Now there is adjustment of two types. First is the coarse adjustment and second one is the fine adjustment. Okay, now what is the great difference between the two? In the core adjustment, it is bigger size screw, and if we talk about fine adjustment, it is smaller size screw. It is responsible for doing the up and down movement of the screw. Okay, up and down movement of the lenses that are fixed there in the body tube. And the fine adjustment is for fine focusing. Okay, did you understand this part? Moving on towards the next part is the reflecting mirror. This is the important part of the microscope, and without it, no cell organelle or nothing can be seen under the microscope. Okay, now if we talk about reflecting mirror, it is meant for reflecting the light rays. It is meant for reflecting the light rays. Okay, now it has written over here also, and light passes through the object which is to be seen. Means it reflects the light that is coming out from the surroundings, and the light passes from the object which has to be observed under the microscope. So these are the parts of the microscope which are responsible for the complete instrument formation of a microscope. Now we are going to read in the next slide that how the compound microscope looks like. So this is the structure of the compound microscope represented over here. So you can see, now we can see the adjustments we have seen over here. The things which we have read over here. First part is the base. Okay, this is the base part on which the whole weight of the microscope has been put. Now next part is the stage part. The stage part is this. Okay, you can see it over here. This is the stage. Okay, this is the stage over here. This is the second part. Now third part is the stage clips. The stage clips are put over here. You can see here, these are the metallic clips which are used to fix the slide over here, which we have prepared for the observation of any kind of cell organelle or a cell. Okay, so these are the stage clips. These are of a metallic substance. Now if we move on, then we can see here the condenser. This is the condenser part over here. Okay, the objective lenses are put on this nose piece. This is the nose piece over here on which these lenses are put. They have a fine focusing over the object. We can change the lenses according to the slide. If we want to observe more microscopic slide or we want to observe more clearly, then we have to put the higher power lens. And if we want to see it by less diversion, then we can see it by a less power lens. Okay, so these are the 2 to 3 types of lenses attached over on this revolving nose piece. So this is known as the body of the microscope. Okay, now where the slide has to be put under observation. This is a great information. So here is the slide put. Okay, now I am drawing it with another pen. It is with yellow colored pen. Here is the slide put under the 2 metallic strips. So this is known as what? Aperture. Aperture is a position on which the slide has to be put. Okay, the glass slide which has to be put. Now you can see here, for the focusing, fine focus knob and coarse focus knob are present. Means it is a adjustable screw type. Okay, we can adjust the microscope according to it. It is for fine focusing and it is for coarse focusing. Did you understand this thing? Now there is a diaphragm and an illuminator. Here it is diaphragm. It is also known as iris. The lower part of this slab, it is known as iris. And this is the illuminator. This is the illuminator. Converges all the light from the surrounding, and we can see the object which is placed on the glass slide clearly because of this. Okay, so did you understand the structure of the microscope over here? And it is very necessary because it is the basic instrument through which all the cell organelles of a cell are being observed. Okay, Now moving on towards the next slide, we are going to prepare a microscopic slide over here. Now what the things we need to prepare the microscopic slide, what are the basic instruments we use to make it? So we are going to study it over here. So first basic thing we use is the microscopic glass slide. You can see it here, students. This is the glass slide. Okay, we take and we put the cell material, as if for an example we have taken a part of cheek cells or onion peel. We put it over here at the center. Okay, then we put a glass slide over it and observe it under the microscope so that we can see the arrangement of the cells clearly. So this is the glass slide, which is the basic component. The second component we use is glass slide cover. This is very important because if we are observing the glass slide under the microscope, it should not possess any kind of air droplets or impurities on it. So to remove these particles, we use the glass slide cover. If we cover the cell or any part which we are taking in observation, it is not going to take up any kind of impurity with it or air bubbles inside it. So it can be clearly visible under the microscope. So these are the small glass slides you can see over here. Okay, and it is put on the glass slide. Okay, these glass slide covers, this is known as glass slide covers, and this is put on the glass slide. Okay, if any object is placed over here and it is placed over the object. Okay, remember this thing. I will show this thing through a diagram and also a video over here. So it will be clearly visualized to you all. Okay, now moving on towards the next component we use in the microscopic observations is for preparing of slide is the dropper, through which we drop the chemicals like glycerin, safranin, iodine, etc. to stain the material which we are using. And the last thing we use is the blotting paper. Filter paper also we can use here if we do not have blotting paper. Now what is the functions of both of these? The dropper is used for putting the chemicals over the slide over here, and this paper is used to remove the extra material or the water or the chemical which is present over the slide. Okay. Now we are going to make a particular microscopic slide. So first you have to know this thing that the object which is viewed under the microscope is known as specimen. Okay, so it is also written over here, the thing which is or the object which is viewed under microscope is known as what? Specimen. And the thin sheets of glass are known as microscopic slides. Okay, now a second much thinner glass which is used to cover the microscopic slide over here is known as cover slip. The very important thing over here is the note. Please put a star mark over here. Knoll and Rushka were the two German scientists who discovered the electron microscope. Now there is a difference between a compound microscope and an electron microscope that you are going to study in your higher classes. Okay, now you have to study about the compound microscope only. So Knoll and Rushka were the two scientists, remember students, in 1932 they discovered the electron microscope. Each and every small particle can be seen under the electron microscope. It can be seen in the compound microscope as well, but we need a great diverging lenses for it. Okay, great power lenses for it. So now we are going to prepare a glass slide. So how do we prepare a glass slide to see under a microscope? These are the basic steps you should follow before preparing any kind of glass slide of any cell, organic or a plant cell or animal cell, anything. Okay, so first you have to take a glass slide over here. Okay, now the next step, this is the step number 1. Take a glass slide, clean glass slide. You should remember this thing. No impurity, no chemical should be over there on the glass slide. Now the step 2, with a dropper, you should put the chemical or the water in the middle of the glass slide. Okay. Now, what is the function of dropper over here? You should put the small drop of water in the middle of the glass slide. The third step for the glass slide preparation is gently put the object to be observed on the glass slide. Okay. Now, you should put the object under the glass slide with the help of a brush. Remember student, you should use a brush over here, or you can use a needle or a forceps over here. Do not use your fingers over here because fingers may contain impurities, fingers contains germs also. Okay, so we should use a brush. Now, the fourth step for preparing a glass slide is objects if transparent are first stained. If you take a peel of onion, you will make understand in the next slide that how do you make a glass slide of onion peel. So I am just giving a sample over here. If the peel is transparent or anything, any cell part is transparent, so it should be stained. So it is the basic step and the fourth step. Now next move on towards the fifth step. The fifth step is hold the cover slip over the object in such a way that no air droplets remain inside the cell. Okay, now if you have put, see it clearly here, this is the glass slide and you have put the object over here. Okay, this is the cell part, any cell part which you are going to take. Now you are putting a cover slip over here, so you should have to place it very carefully so that it touches the edge of the drop of water and you gently lower the cover slip over the water so that extra amount of water will get removed from it and no air bubbles are present under the observation. Now this is the basic fifth step. Did you understand this? The last step and the sixth step is you should dry the extra amount of water with the help of a filter paper or a blotting paper. Okay. Now, see it is written over here also, with the blotting paper you should dry extra amount of water. And you should take care of the slide that it is clean and dry. Okay. You should not place the wet slide under the microscopic observation. You should know this thing. We always put a dry slide under the observation, the specimen which we have prepared. So did you understand all the all the 6 steps of preparing a slide? Okay. Now moving on towards the next slide. This is a small activity. In this activity, we are going to make a slide of onion peel. Now, how the cells are observed in a plant cell, we have to see this. As you know, the plant cell has cell wall also, cell membrane also, and many more cell organelles also. So, it can be seen under the microscope very clearly. And the basic fact, that is the vacuole, the large part that covers the plant cell, can be seen under the microscopic observation. So now let's prepare the slide. Okay, the first thing which we are going to take is the onion peel or real leaf cell. Now the materials which we are going to be required for the preparation will be onion, glass slide, cover slip, stain and microscope. Here stain means the chemicals like glycerin, iodine, saffron, etc. Now the next step will be the procedure. Now what we have to do it, we have to cut the onion slice into 2 pieces. We have to peel out the middle layer with the help of forceps and then we have to put that middle layer on the glass slide. Now we have to use the chemical, that is the stain, by a dropper. We have to put only 2 to 3 drops of that chemical on that cell part. Okay, that peel part which we have taken on the glass slide. Now what we have to do, we have to take a cover slip and we have to put it gently over it so that no air bubbles occur over it. Now what will happen, a glass slide is now prepared as a specimen. You should put it under a microscope, fix it with the clips which are present, the metallic clips which I told you about, fix it with that and then you should set up the lens over the coarse adjustment. If you will set up the coarse adjustment and the reflective index of that glass, the condenser and everything, you will see that the clearly plant cells are being observed, in which you can see the cell wall, the cell membrane, the vacuole part, and even if you have made the slide clearly, then you can see the plastered over it also. So these steps are written in the procedure. You can read and note it down in your notebooks. Now, what are the observations? We have to see it. So the observations will be a number of cells are present side by side. What do you mean by side by side, students? If there is a cell over here, okay, there is a cell wall, there is a cell membrane, and there are small vacuole, a large size vacuole, sorry, and small organelles are present over it, then it lies besides it. Means the side by side, the cell wall and the cell membrane are present. The middle lamellae will be present. Okay. Everything which I told you about will be present. So it is the vacuole which is going to be observed, the cell wall, the cell membrane, and many more organelles, or we can count them into cytoplasm of the cell. By writing its cytoplasm, we cover all the cell organelles present inside the cell. Okay, so you can see here the distinct wall that is called the cell wall can be seen. Center of each cell there is a nucleus. The nucleus can be seen, and the large empty spaces exist within the cell. These are known as vacuole. So the large vacuoles can be seen inside the plant cell. And we have to see a particular video to understand how the staining takes place, how the glass slide is prepared, and how every procedure is to be carried out. Okay, so let's start with our procedure of making a slide of an onion peel. So the first thing you have to do, what you have to take, that is the materials required for making the glass slide. So first you have to see the slide over here, the minimum sized onion over here, cover slip over here. Okay, you can see in this video the forceps are also available. I told you you should not use your fingers for putting the cell part over the glass slide. You have to use the brush or the forceps. The glycerin, dropper, everything which I told you, every component is necessary for making a glass slide. Saffron in solution for staining, and a compound microscope, last part, is present over here. A brush is necessary, you know that. Okay, now what is the procedure to make the glass slide of an onion peel? First you have to take a middle size onion and you have to cut it into 2 pieces. So it will be cut into 2 pieces. Now what you have to do, you have to separate a layer which is transparent over here. When you cut an onion, you can see that there is a transparent layer present in the onion. Okay, so this part you can see it clearly. A transparent layer is present over here. Okay. Now what it is going to happen, this part is going to be taken into the slide. Okay, with the help of forceps, not with fingers. Remember this thing, you should not use fingers in any kind of glass slide making. Okay, now it is staining with the water and then with the saffronin to make it germ free and to make it removing from the impurities. Okay, now with the help of brush, after 2 to 3 minutes, it is going to be put over the glass slide. Why we are taking 2 to 3 minutes over here? Because if the germs or impurities are present, they are going to be removed it. Now what will happen, students? We are taking a glass slide over here, putting a drop of glycerin over on the glass slide. Okay, now we are putting the cell part, that is the part which we have taken, the transparent part, through a brush. After putting that part over the glass slide, it is going to be observed that we are putting a cover slip over here. Okay, now by putting the cover slip over here, we are going to use a blotting paper, and by pressing it with needle, we see that the air spaces or the air holes which are present inside it are going to be removed, and the extra amount of water are removed through a blotting paper. So the slide will become dry and clear. It is necessary condition. Now we are observing it through a microscope. You can see here the cell part. It is very clear. Okay. Now in this cell part, you can see here the cell wall is present, cell membrane is present, nucleus is present, and the last part, cytoplasm, can be seen. The cell wall, cell membrane, nucleus, and cytoplasm, and these have a very distinct vacuole part also, which is present over here. Okay, large size vacuoles are present in plant cells. You know this thing very clearly. Okay, so this is the part how we can stain the slide and make the slide and observe it under the microscope. So this is the whole procedure to make any kind of slide under the microscopes. Now did you understand each and everything? Now we are moving on towards the next slide. This is the basic step and the procedures written over here. Okay, now we are going to revise this thing which we read in the video. First we have taken an onion and put it into two quarters. Then we have removed the fleshy scale leaves, and in the third step we are snapping the leaf backward. And in the third step, what we are doing, we are taking the transparent material out of it with the help of a forceps. Now we are putting in this inner layer of the epidermis. Okay, it is peeled off from the onion part. Now we have to put this on the slide. Epidermis is placed on the slide, covered with 2 to 3 drops of distilled water. Okay, now cover slip is lowered over it, means it is put gently over it. Okay. Now the sixth step, drop of stain is put at one end of the slide, and in the last part what we have to do, the stain is drawn over the specimen using a small piece of filter paper. You can use here filter paper, and blotting paper also can be used to remove the extra amount of water which is present over the slide. So the slide will become dry and clean. Okay. Now you have understood all about how the microscopic slide is to be made and what are its procedure, what are the basic components we use to make a microscopic slide. Now this is a great observation which we have seen of the plant cell. I am going to label it again. You have to revise each and everything, students, because it is very necessary. So first part, you see here these distinct walls are created over each and every cell. Okay, it is clearly visible in this diagram over here that each and every part is to be distinct here. Now this is the nucleus over here. The nucleus in each and every cell can be seen clearly. This outer part which is to be seen clearly is the cell wall, and besides it the cell membrane resides. Okay, besides it the cell membrane resides. So this is the cell membrane. The part which is present inside a particular cell is known as what? Cytoplasm of the cell. Okay, and the large size vacuoles are also present in it. So we can label it here. And these, every cell is attached by another cell. You can see here the arrangement is like a brick arrangement. Okay, if we put a brick over one another, you can see this kind of arrangement. Okay, so in the plant cell, this type of arrangement of cells is present. Okay, now you have seen it clearly how the cell wall and cell membrane are being distinguished. Where is the nucleus present inside the cell? Every plant cell contains this type of cell arrangement. Okay, now moving on towards the next part, we are going to study about the video of cell organelles. That is the most interesting part of today's lecture. So we are going to observe many cell organelles like chloroplast, like endoplasmic reticulum, lysosome, and even the mitosis phases which we have read in lecture number 6. It is very important, students, to read these kind of things through video lectures, because you can understand this thing clearly, that what is going to be happen inside the cell organelle. As you read about protein synthesis, lipid synthesis, transcription, translation, cell division, and many more things you have read till now in the lectures. So we are going to observe these kind of things in this lecture through videos, and we are going to see how the things is to be synthesized, how the things are prepared inside particular cell organelle. So let us start with the first cell organelle, that is chloroplast with us. So first we have to see about the chloroplast. Before starting the video, I am going to tell you a small little bit things about chloroplast. What was chloroplast, students? Chloroplast is the chlorophyll pigment which is used for the photosynthesis process, which is represented by the plants to perform the function of photosynthesis and to make their own food. Okay, so you know it very well. Now I am going to start the video of the chloroplast, and you have to observe the basic two components of the chloroplast. You know this thing? Have you not forget? And you should not forget these things because we have read it very carefully. Okay, so chloroplast contains two basic things, that is the grana and the thylakoid. You know this thing? The thylakoid are stacked over one another to form grana. Okay, and these are interconnected through the intermembrane. Okay, now they perform the functions of photosynthesis by the systems PS2 and PS1. And the main thing they synthesize through these procedures are the ATP, that is adenosine triphosphate, and NADPH, nicotinamide adenosine dinucleotide phosphate. So let us see with the video that how the chloroplast works. So this represents the chloroplast over here. Now you can see in this diagram clearly, students, that first thing we can see here that I told you about, chloroplast is present in different sizes and different forms. In Chlamydomonas, it is present in U-shaped, I told you, and less in number means their numbers are fixed in Chlamydomonas and in Euglena. If we talk about the plant cells that are higher plants, the chloroplast numbers varies accordingly. Now you can see here they varies in the size. They are up to 4 to 5 micrometer in diameter. Now you can see here the structure clearly. If we talk about Chlamydomonas, they are cup shaped. If we talk about Spirogyra, they are ribbon shaped. Spherical in shape in higher plants. And if you talk about a particular plant cell, where are the chloroplasts located and what are their basic components? So I told you about the grana and the thylakoids in this. So where are the grana and the thylakoids situated over here? You can see it as a double membranous structure. So first is the outer membrane, second is the inner membrane, which is separated by the intermembrane space. So you can see here, students, this is the outer membrane, this is the inner membrane, and this space which is present between the outer and inner is known as the intermembrane. Now you can see here the ribosomes are embedded in the stroma, means in stroma the ribosomes and the DNA as well is present. And because of that, I have said that it is semi-autonomous cell organelle. Semi-autonomous cell organelle means which contains its own proteins, ribosomes and DNA. Means it can self-replicate. It can make a replica of itself. Okay. Now, you can see here, this structure is known as what? Ribosome. And this structure is known as what? DNA. Did you understand this thing here? This is seen clearly in this diagram over here. So it is known as semi-autonomous. Now this structure is known as grana and these are known as fret membranes. Okay. Now the integral lamella. What is the intergranal lamella? Intergranal lamella is this structure which connects the two grana by them. Okay. Now, you have known very well that photosystem 1, photosystem 2, ATP, NADPH and everything else which is synthesized inside it. Now, the photosystem 1 and photosystem 2 helps for the light reaction which we have read in the lecture number 6 as well. Now, you can know this thing that in this, The ATP synthase is present over here, which is represented by the orange color pigment, and photosystem 1 and photosystem 2 are also represented over here. Now photosystem 2 is bigger in size and the photosystem 1 is smaller in size. Why this is so? Because electron transport chain, that is the gradients which are larger of the photosystem 2, and in the photosystem 1 they are smaller, means there is only a single pigment known as ferredoxin. And in the photosystem 2 there were three components or the three electron transport chain acceptors, that were the plastoquinone, the cytochrome, and the last one was the plastocyanin. Okay, plasma membrane. Now I am going to explain you about the plasma membrane, the fluid mosaic model of the plasma membrane. Now what was the fluid mosaic model? Who discovered it? Did you remember this thing? Singer and Nicholson were the two scientists who were responsible for development of fluid mosaic model. Okay, now it is the plasma membrane. Now you can see here the structure of the plasma membrane, the fluid mosaic model. Now we have read about the lipid bilayer, and the lipid bilayer is what? The phospholipid layer which has been stacked over one another. There were two phobes, one is the hydrophobic part and the hydrophilic part. The hydrophobic part remains inside and the hydrophilic part remains outside. Now you can see here this type of arrangement is there, means one hydrophobic tail is fixed with the another hydrophobic tail, and this kind of arrangement is the lipid bilayer arrangement of the plasma membrane. Okay, so you can see here this type of layer is formed when these lipid molecules are arranged over here. Now there is the focusing of proteins over here. I told you about the proteins that are embedded between the lipid bilayers. So it may be peripheral proteins, it may be globular proteins, it may be quaternary proteins, any type of proteins are embedded over here. So these you can see it, the green color pigment which is moving in this video, it is the protein which is embedded between the layers of the lipids. Okay, now this is the end of the plasma membrane and the fluid mosaic model. Now we are going to discuss about the another cell organelle, that is the endoplasmic reticulum, and it is also known as ER in the short abbreviated form. Okay, this is my favorite cell organelle because its diagram is too easy to learn, and the basic thing which is present inside our cell is endoplasmic reticulum. So we should start the video over here. So in this cell you can see here there is nucleus present over here, and around nucleus the endoplasmic reticulum is scattered and it is a large surface area. Now you can see in bacterial cells endoplasmic reticulum is absent. Okay, you have to learn this thing. Now if you observe here, the endoplasmic reticulum is scattered over the cell part in the cell, in the cytoplasm of the cell, okay, near the nucleus. Now there are two types of endoplasmic reticulum, one is the rough and another one is the smooth. Now rough endoplasmic reticulum is because of the presence of ribosomes over it, and smooth does not contain ribosomes over it. Now you can see here these are the small globular structure, these are ribosomes. They do the function of protein synthesis. Okay, now in this diagram you can observe here that these are the endoplasmic reticulum which are present near the nucleus is the rough, and these which are present here, it is the smooth endoplasmic reticulum. Okay, now smooth endoplasmic reticulum is scattered in a large surface area over here near the rough endoplasmic reticulum. Now what are the functions of both? We have read it clearly. Because of the presence of ribosomes, the protein synthesis takes place here in the rough endoplasmic reticulum. Now the lipids are the molecules which contain what? Hydrocarbons, and make up the building blocks of the structure and function of a living organism. Like lipids, fats, anything, oils are synthesized over here. Now what happens, students? This smooth endoplasmic reticulum helps a lot to the vesicles. Okay, the protein content or the content like oils, lipids which are going to be synthesized are going to be collected inside the SER, that is the smooth endoplasmic reticulum. So it helps a lot for the functioning of a cell, or we can say that it is the basic part which is attached with the nuclear membrane. Okay, now did you understand about the endoplasmic reticulum? Moving on towards the next part is the lysosome. Now, if we talk about the lysosome, what are the lysosome? It is the small structure which does not have fixed shape. Now, what happens? These are the lysosomes which contains usually hydrolytic enzyme. I told you about this thing in the past lectures. So, hydrolytic enzyme, what are their functions? Hydrolytic enzyme activates the secondary lysosome activates the hydrolytic enzyme, and then they are passed to the residual bodies, and the unwanted materials which is to be carried out through it are thrown out of the cell. Okay, so it is like the refinery of the cell. You can say it like that because they get ruptured and release the enzymes, or we can say that they are helpful in removing the unwanted material of the cell and maintaining the waste part out of the cell. Okay, so you can see here these are transported from uh, vesicles to the mitochondria. The important part are being transported from vesicles to the mitochondria, and the unwanted part are removed through residual bodies. And if the cell is in need of any kind of enzyme, then what happens, students? The autophagosomes or the autolysosome ruptured and releases the enzymes which are helpful for the cell. Okay, the cell functioning. So it is all about lysosome over here. Let's next move on to our next part, that is the ribosome. Now, what are ribosome? You know it very well. They are the protein factories of our cell. So how does they manage the protein synthesis? So we are going to see it through this video. So what happens, students? These are the ribosomes which are embedded on the surface of the endoplasmic reticulum. You can see here these purple colored ribosomes are embedded on this structure. Okay, now what happens in this cell? Many ribosomes are present for the protein synthesis. Okay, now how the protein synthesis takes place? You know it very well through transcription and through translation. The gene template which is present in the DNA has to be get synthesized into mRNA, and this mRNA, when is prepared, is translated, and it is ready for the translation process and read by ribosome. So what happens? The polypeptide chains are formed. I told you about the peptide chains, the amino acid, in the last lecture. Did you know the ribosome has two small units? First is the larger subunit and the smaller subunit. So the mRNA is being bounded between both of them. Now the coding is to be used here. Why the code word is used here? Because of the presence of amino acids through which the proteins are to be synthesized. Okay, now it is written over here the ribosomal functions which are to be represented here. A ribosome which functions like it is protein synthesis. The CUG which is seen over here, this is the cytosine, uracil and guanine. These are the amino acids which are responsible for the protein synthesis. These amino acids form a chain, and this chain is get to be repeated again and again, and the protein synthesis takes place. You can see it over here. Now ribosome is saying, voha, I have completed all the procedures of protein synthesis. Now what happens? They use some enzyme and the exocytosis takes place. Exocytosis means the extra enzymes are to be removed and the useful enzymes are to be used. Let's move on towards our last thing, that is the mitosis. Okay, now what is mitosis, students? It is the basic part of the cell division through which a parent cell divides to form daughter cells. A parent cell divides to form what? Daughter cells. Okay, now how the daughter cells are formed? And actually mitosis is the basic part of a living organism itself. If we talk about reproductive processes, how the zygote is formed, how the cell division takes place in a particular zygote and it turns into a big organism through a small cell. Okay, now this is a great mystery here, students, that how a small singular cell gets divided to form multiple cell and a multicellular organism takes place birth. Okay, so we should see it through a video representation and we should understand how the reproductive things takes place and how the mitosis takes place. In the last lecture, we talked about the cell division, the phases of the cell division, the mitotic division, that is what? The interphase, G1 phase, S phase, G2 phase, the development of the cell, and then how the cell divides and makes its replica, and how the diploid cell again divide to form the diploid cell. Okay, now the basic phases for this thing will be what? The basic four things which we have taught. The basic four phases which are represented over here will be all the phases like the prophase, the metaphase, the telophase, the anaphase. Okay, now step by step we are going to see how the chromatin gets condensed. They form chromosomes, and these chromosomes, when they are matured, they form the spindle fibers, and the centrosome helps in that. Now how they are attached, they move towards the equator, then they move towards the upper part and they get separated. The sister chromatid gets separated, forming a new cell organelle or a new cell. Okay, now we are going to see it through a video over here. So let us start with the cell cycle, how the cells divide. So a particular organism grow. We are the little baby when we take birth. Now this little baby grows and forms a particular adult. First we suffer from a juvenile stage and then we go to the adult stage. Now what happens in this, students? These cells divide. Okay, the karyokinesis and the cytokinesis takes place here. Now what is this? The cytokinesis is the division of the cytoplasm and the karyokinesis is the division of the cells. Now what happens, students, in this procedure? First the DNA replication takes place over here. This is the basic step in which the DNA replicates. Now how the DNA replicates, what happens? These are the stone proteins which are present inside it. Okay, now this is a basic chromosome which is formed by a pair of chromatid. I told you in the last lecture also. Now this is the chromatin material, you can see it over here, and after this is the chromosome. Okay, this is the center part of the chromosome and these are known as sister chromatids. Why they are known as sister chromatids? Because two chromatids make a pair and they are known as chromosome. Now in mitotic cycle you can see here G1 phase, S phase, G2 phase, and last one is the mitotic phase from which it occurs. Now the phases that occurs in the mitotic division are the main phases. So first one is the G1 phase, that is the gap 1 phase, in which what happens, these centrioles help them, these cells, and next is the synthesis phase, that is the S phase, in which the cell organelles try to perform their functions which have been developed in the G1 phase, and the last phase is the G2 phase, in which what happens, students, the DNA are ready to replicate over here. Okay, now the mitotic division takes place in 4 phases. What are the 4 phases over here? First phase which occurs is the prophase, then metaphase, then anaphase, and last one is the telophase. So first is the prophase. What happens in the prophase? I told you the chromatin gets condensed and forms what? Chromosome, and a centromere is the basic thing which attaches both the sister chromatids. Now this chromosome formation has been taken place in the first stage. Now the prophase, in this phase only this happens, the centrosome comes here over it and start forming the small fibers to get attached toward the sister chromatids. Now you can see here these are getting attached over here. Okay, and this is the kinetochore, which is the protein, the basic protein which helps in attaching them. Okay, now in the metaphase stage they comes to the equatorial position. I have told you this thing that they come in the equatorial position, means in the tilted position they have come here. Okay, you can see here the tilted position, equatorial. Now in anaphase stage what happens, they try to separate, the sister chromatids get separated. Okay, so this sister chromatids get separated over here. You can see here the contractile ring is formed and this is going to be break in the last step. Okay, now this you can see here in the telophase stage they are going to break and they are going to be separated. So this process happens in the telophase stage. Okay, these spindle fibers are broken, means the work of the centromere is now over here, means the centrosome which was working from the first phase, that is prophase, is now over here. Now what happens, the cytokinesis takes place here, means the cytoplasm gets separated of both of the two cells which are formed, the daughter cells which are formed over here. You can see it through a diagram that they are getting a diversion over here. The cell plate is being formed between the two cells. Okay, now these cells are getting separated from both of the two, means they are being separated completely. Okay, now these are the basic step. After G1, S phase occurs, after S, G2 occurs, then mitosis, and last procedure is cytokinesis, which is not counted in the phases. It is just the basic step in which the cytoplasm of both the cells get separated, and both these cells now can survive independently, means they do not need the cytoplasm of another cell to get survive. Okay, now these are the phases, you have to learn it. Okay, this is G1, this is S, this is G2, the mitosis, and last one is the cytokinesis. Okay, these are the checkpoints. There are various checkpoints that the cell is particularly working or not. These checkpoints are necessary if the cell organelles are getting developed in G phase. So in S phase, they are checked that how the cell organelles are working or not. Okay, if they are not working or they are not performing their functions, the cell is turned to be damaged and thrown out of the body. Okay, so this is the nutritional state of the cell, which is to be checked by itself only. Now you can see the mitotic procedure clearly over here. The G1 phase, the S phase, G2 phase, and the mitotic divisions which occurs. Okay, the chromosomes are being divided, the spindle fibers are being separated, cytokinesis takes place here, and the cytoplasm is being divided. So you can see here this cytoplasm is being divided and the new cells are being formed over here. So this is how the whole procedure of cell division takes place. Okay, now I am ending my topic, and the cell part, that is the fundamental unit of life, the chapter is being completed. You should solve the module, and in the next lecture we are going to discuss about the module discussion. Okay students, thank you.